Okay, on the screen right now, here I have a coronavirus. And you see all those things on the surface of it? They are what we call spike proteins. They are the very things that allow the virus to be able to latch on to our cells. Once they latch on like a hook, that's when they will invade us. So scientists have been trying very hard to find a way to destroy these hooks. Once if we destroy them, then they cannot cling onto us anymore. If we were to zoom into one of these proteins, this is what the protein looks like. Can you tell me what levels of structure are present in this protein? Okay, please discuss with your friend. Have a conversation right now. Yes. This is how you normally talk to people. No. Hey, where you go? What do you know? What do you know? Okay, what levels of structure do you observe in this protein? Maybe it's not observable. Okay. Three, yeah? Okay, so there are 3 levels we learned, right? This protein has which levels? Sorry? All 3. Okay, let's elaborate. If you say all 3, what's the first level of structure called? Primary. Could I get someone to describe and elaborate what it means by a primary structure? Michelle? When I say a protein as a primary structure, what am I actually saying? Sorry? You try to look through your notes. Okay, everyone look through also. What is a primary structure? How will you describe a primary structure? Michelle, once you got it there, ask her. Sorry, say again, louder. Okay, so thank you. Primary structure is simply describing the number, sequence of the amino acids. And perhaps you can include also one more layer, the type. Okay? At the end of the day, the number, sequence, and type of amino acids determines the protein's primary structure. Every protein has a different primary structure. Every protein, or in other words, every protein has a different number, sequence, and type of amino acids in its chain. Okay? So if I were to draw out a primary structure without secondary structure or without tertiary structure, it will just look like a string of beads, except the beads are your amino acids. What are the bonds holding the amino acids together called? Sorry, louder. AO3. I know all of you all know the answer, and we need to have a conversation now. Yeah? When I ask something, I do hope that you all respond and work together. Okay, I did hear one person say it, but I don't believe only one person will say it. Yeah? We need to work together. Okay? Right now, I feel like I'm really, every time I turn on chapter, I always go wrong. Okay? So, the bond that holds all the amino acids together is a peptide bond. Shreya said that there are 3 levels of structure. The next level is the secondary structure. Okay, then now very quickly everyone look to the screen. What types of secondary structure do you observe here? Okay, thank you. Yeah? You can see alpha helix. Usually that's the most obvious. It's like these curls over here. Okay, so we observe alpha helix. You can write it as this alpha helix. Okay. You see beta pleated sheets? See? Okay, over here there's some, over here there's some, over here there's some, over here there's some. Beta pleated sheets. If I were to now take this primary structure and introduce some secondary structures, you'll end up looking like this. From a straight chain to now, let's introduce one alpha helix. Let's introduce one beta pleated sheet. Let's perhaps introduce another alpha helix. Okay? Notice it has not folded in a very convoluted way yet to form this globular shape. Yeah? Which brings us to our final structure, tertiary structure. The tertiary structure, what defines the tertiary structure is that it is highly folded to form a 3D globular shape. Like, in other words, a lump. Okay, it's a lump. So let's fold this even further. If I take this primary structure with some secondary structures inside and fold it even further, I will get a lump. Okay, maybe here's the alpha helix. Yeah, bend a bit more. Let's introduce the beta pleated sheet. And then let me bend a bit more, and then I introduce the last alpha helix. It is now a very compact 3D globular shape. Or another way to see it is, if I just draw a series of squiggles like that, this is just a tertiary structure and a primary structure. No secondary structures present. Yeah? Okay, so maybe I put this out. This one has number 1, number 2, number 3. This one only has number 1, number 3. This has a primary and a secondary structure. This is just a primary structure. If I were to do this exact same thing now, hey, I have more whiteboard space now. Okay. So let me do this same squiggle. Okay, there's a primary and there's a tertiary structure. If I wanted to, let me introduce some secondary structures in red. Okay, I'm going to erase this part over here. Let me introduce a alpha helix. Perhaps over here, I will introduce a beta pleated sheet. Okay, and over here, let me introduce another helix. Alright, so now I have also secondary structure. Okay, the skill to pick up really is we'll just show you a protein. You tell me what levels you observe. Okay, tell me what levels you observe and also be able to describe. Okay, so you must be able to tell me the levels, and you must be able to describe the levels that you see. Go back to that simulation that we played with, that I've shown you before. Okay, we know that proteins fold of two things. The medium it is in, and also the interactions of bonds holding the protein together. So let's focus instead. Let's say we're in a vacuum, right? If I were to click play in a vacuum, you'll find that they'll still come together. What's holding them together? Lots of interactions and bonds. So if at the primary structure we say that the amino acids are held by peptide bonds, tertiary structure, we're going to draw dotted lines to represent all the bonds holding this complex 3D shape together. These bonds include a variety of things as we described yesterday. Yeah, I'm going to draw the bonds in red. Okay, the kind of bonds you can look at are, number one, ionic bonds. Number two, hydrogen bonds. Number three, hydrophobic interactions. Last but not least, covalent bonds, but specifically only one kind of covalent bond called disulfide linkage. Okay, 1, 2, 3, and 4. 4 kinds of bonds that can hold this complex shape together. So that, even if I take it out of water or oil, it will still hold together. You'll be very sad if all that's holding this in this shape is the water surrounding it. Then when you're dehydrated, you really start to crumble and fall apart. You're still held together, right? So fundamentally, there's still some bonds holding together. Okay, welcome back. Okay, then how?
I'm going to.
I see. So you're replace. Okay. Yeah, so that's a recap. Okay, so we've done just now a quick recap. Today we're going to talk about something new. Let's look at the secondary structure a little bit deeper. Because fundamentally, something is, the peptide bonds are holding the amino acids together. These 4 kinds of bonds are holding the tertiary structure together. But what in the world is holding this helix in this shape? What in the world is holding this beta-beta shape in this shape? Why doesn't it just unfold? What's keeping it held in this folded pattern? I'd like you to look at your class activity. If you scroll to the second part, okay, part D of your class activity, CA 1.5C, you will see on the left hand side beta-beta sheet, except zoomed in so you can see all the atoms. On the right side, you see an alpha helix, except zoomed in so they can see all the atoms present. So could I get you to label beta-beta sheet, alpha helix? Okay, I may provide you this zoomed in version. Do you see all the C double bond O and the NH? Yeah? Okay. Can you just highlight one of them? C double bond O and NH? Because what that represents are all the peptide bonds in between the amino acids. So if I were to zoom in, you see a C double bond O, you see a NH, that is a peptide bond. So there are lots of peptide bonds here. Even even in an alpha helix, you can observe lots of peptide bonds. Could you highlight one peptide bond in the alpha helix? Find a CO and an NH, and then you highlight them together. That's a peptide bond. Okay, so find an example of a peptide bond in the beta-pleated sheet and inside the alpha helix. Because we're going to look at how they come into play to hold these secondary structure together in a shape. Who give you one? What, you wrote this yourself? The O writes this, alpha is too amazing. Does that explain? Okay, sure. It's to hang there. One day you see me coming with a Visitan is too amazing hanging around my neck. Okay? Yes, Henry?
So the peptide bond is between the amino acid, right?
Yeah.
Then the 1, 2, 3, 4 there, right?
Yeah.
It's between
What is it between?
the primary structures.
Okay, so we hold on. Okay, so let me ask an interesting question. All these bonds, when I draw these dotted lines, right, what exactly is it between? That's a great question. What is it between? Yeah, it's the R group. So, can you see over here? The R group over here, the R group over here. Yeah. So, all these interactions we see over here, if I were to zoom in, so let me just zoom into one of them. Let's zoom into this section over here. What you will observe is that there's a carbon, there's an R group. Okay, there's another R group, there's another carbon. And then interaction is taking place between them. Okay, it's between R groups. So thanks Henry for that question. This is bonds / interaction between R groups. What's holding the alpha helix and beta pleated sheets in this formation? Okay, the bond that is involved that's holding all these fellas together is hydrogen bond. We heard hydrogen bond before, yeah? Okay, the hydrogen bonds are occurring within the chain. It is holding this alpha helix shape together, beta pleated sheets also. Hydrogen bonds are holding them in this conformation. So if I were to point the arrow, the bonds holding each structure together are hydrogen bonds. So let's do a quick practice. If you look at this beta pleated sheet going down and then going up, can you draw in dotted lines to represent where the hydrogen bonds could take place to hold these two sheets together? Where can hydrogen bonds take place within this alpha helix to hold the entire helix together so it doesn't unfold? Okay, I'd like you to use dotted lines now to draw in where you think hydrogen bonds can take place. And then we'll go through in a while. You can work with each other. Please work with each other. Identify where you think the hydrogen bonds can take place. Let's see. It's a man. Please. Yes. haven't drawn the hydrogen. You have to draw the hydrogen bond. Okay, what will you need to be able to hydrogen bond? Correct? And you also need to be able to. Oh, you're applying chemistry, is it? Okay, hydrogen bond. Okay, maybe I do a quick recap of hydrogen bond. So to perform a hydrogen bonding, you need to find number one. Okay, let's just do it with water. Okay, let's just do it with water. So imagine this H2O. Okay, this H plus. Sorry, this is a plus, this is a minus. Right? And let's say I have another polar group nearby. Maybe that polar group is NH2. Okay? Hydrogen bond will occur between the H and the N over here. Yeah? So the skill to apply here is, can you find out where all the partially negative atoms are, where the partially positive ones are, and then you draw hydrogen bonds to hold this structure and this structure together. Yeah, I'll go around to have a look at what you've done. Bond these two together. Now I'm asking, what is holding? Where can you form bonds within this group? Where can you form bonds within this group? Yeah, within the What? Yeah. Oh, it really depends on, because it could be ionic, I don't know because we just got Yes.
Both the ionic, like all those bonds and bonds for
To form a tertiary structure.
so both of them are like the same,
All four, you mean?
like ionic hydrogen, like away covalent and
Yeah, all the same function.
they are all the same function to all elements.
Yeah. Just got 4 different ways. Anyway, make sure your hydrogen bond is in dotted lines. Yeah. Can you find the hydrogen bonds